This video goes through events of meiosis I. Before the beginning of meiosis I, there's a synthesis phase, an interphase, in which each chromosome is duplicated. Each chromosome becomes a pair of identical chromatids. At this stage, they're not visible with a light microscope. In prophase I, the nuclear membrane begins to fragment. The chromosomes are what are called tetrads, and I'm designating the blue ovals as the chromosomes that came originally from the sperm, and each pair of ovals is a pair of identical sister chromatids. So I'm drawing a couple genes on each, little a, little b, and the red rectangular chromosomes are from the mother. They're from the egg, and I'm putting big A's, big B's, and big C's, and big D's on those. The color is just to tell them apart. You wouldn't be able to tell just by looking at them. They're connected by the centromeres at the center, and there's a synaptonemal complex holding the whole thing together. Crossover occurs at one or more locations between the homologous chromosomes. So I'm showing crossover between a pair of these, and I'm going to redraw them now to show what happens after crossover. We've shifted all the DNA from one end of one pair of chromatids to the other, and I'm showing now we've got maternal DNA on one of the paternal chromatids and vice versa. And you can see them here with the arrows. There are chiasmata. They're the sites of crossover on each tetrad. It's a protein complex that actually causes crossover. In prophase I, the nuclear membrane at the end has completely fragmented. In metaphase, the tetrads line up on the metaphase plate. And here I'm showing them lined up on the metaphase plate. But it is arbitrary. It's random as to which set of chromosomes faces which side. So I'm just shifting two of them so that you can see that on one side it's mainly maternal, and on the other side it's mainly paternal chromosome. They're connected by spindle fibers from the centromeres to the centrosomes, these little pair of cylindrical structures that organize microtubules. And these are the spindle fibers connected to the centromeres that are going to pull these apart in anaphase. So in anaphase, we're pulling on these, and each pair of chromatids gets moved toward a pole. And I'm erasing the spindle fibers just to get them out of the way, but of course they're pulling, those spindle fibers are pulling the whole time. That's the reason the pairs of chromatids are moving. In telophase I, the chromosomes are now at the two poles of the cell. I'm going to redraw this membrane to show it longer. I'm moving the chromosomes to each end. Spindle fibers are not needed anymore. And a cleavage furrow starts to form. This is a ring of actin with some other proteins that begins to pull in the membrane. So redraw the membranes here partway through cleavage, and then redraw them again. Now we have two separate cells. They are haploids. Some of these chromatids are recombinant. They contain DNA from the mother and from the father. And at this stage, at the end of telophase I, we have two haploid cells, both 1n equals 2.